and then moves on to the somewhat more clinically relevant and easier stuff. So last time we talked about what goes on inside the mitochondria, and I thought I would just put it up on the board in a simpler way so that you'll have a clearer understanding of what I want you to know about all that complex stuff, the citric acid cycle, the electron transport system, all that. So the short version is this. The mitochondria can only use very small carbon fragments. The nutrients in the body are, like glucose, for example, is 6 carbons. But anything that big, the mitochondria can't process. So as we'll see later on today, in order to feed a carbon chain to the mitochondria, it needs to be very small. So like it needs to be a 3-carbon or 2-carbon chain. And then, so those, so a 2-carbon chain goes in to the mitochondria. And the first part, this part right here, this is the citric acid cycle. And there's all kinds of complex chemistry to it, but I'm not interested in you knowing that for this class. You may have to for another class, but not for mine. So the citric acid cycle is this circle right here. So 2 carbons come in, and then we take a 4-carbon chain that's already inside the mitochondria, and we join these 2 together, right? So 2 plus 4 is 6. So we get a 6-carbon chain. Then we start knocking CO2s off of that 6-carbon molecule. So we go from 6 to 5. When we do that, we lose a CO2, right? So where did the missing carbon go? It went and became carbon dioxide, right? So now we have a 5-carbon chain. We do that routine again. We knock another CO2 off. Now we have a 4-carbon chain, and now we've come back to the top of the cycle where we can bring in a 2-carbon chain from the outside again. So this goes round and round and round. 4 plus 2 is 6, minus 1 is 5, minus 1 is 4, and then we add 2 again. So we call it the citric acid cycle because it literally goes round and round and round. Now, so the reason I have the CO2 circle is this is where the CO2 we make comes from. So it comes from this citric acid cycle part. As the CO2s get knocked off, that creates the CO2, which we then have to exhale. All right. So there would be no point in doing this cycle if there wasn't a gain, if there wasn't a benefit somehow. Because making CO2 is not the point, right? That's the side effect. The point of the TCA cycle are these 2, the NADs and the FADs. The mitochondria has a kind of machine that can take hydrogen ions and make ATP using their energy, right? So as this citric acid cycle turns, each turn of the cycle produces 6 hydrogen ions that are attached to an NAD molecule and 2 hydrogen ions that are attached to an FAD molecule. Not important for you to know what NAD and FAD are. Just think of them as hydrogen carriers. They can hold on to a hydrogen. So then those hydrogens enter the second piece of the mitochondrial machine, and that's the electron transport system. So this side is the ETS. This side is the TCA, the citric acid cycle. So all I'm interested in you knowing about the electron transport system is that these hydrogen ions that are freed during the citric acid cycle, one way to think of it is it's sort of like a hill, or if you lift something up and drop it, it falls, right? And that falling motion can be used to do work, turn a wheel, or something like that. So these hydrogen ions essentially fall towards an oxygen radical. So, typically when we talk about oxygen, it's O2, right? Well, you can have an oxygen atom that has, it's 8 electrons. Remember a little bit about that, right? Everybody wants 8 electrons. So this makes it a negatively charged oxygen. This is a very energetic atom. O negative, it does not like to exist like this. So because it's this high energy state, essentially these O negatives pull on hydrogen ions where there are hydrogen ions present. So it's like falling, it's like dropping something heavy. You know, it's the heavy thing is pulled to the earth. Well, the hydrogen ions are pulled towards these oxygen radicals, right? And when they find an oxygen, they make H2O, right? And this energy of falling towards this oxygen radical, it powers the production of ATP. So as these hydrogen ions fall towards the O negative, ADP is added to P, and we get ATP out. So the net result of this whole thing inside the mitochondria is we take a 2-carbon chain, we put it in. As long as there's oxygen present, we get CO2, water, and ATP out. Okay, so that's sort of the overall. So TCA cycle makes CO2 and charges up NADs and FADs. And then NADs and FADs, the hydrogen ions, as they make water, ATP is formed. Okay, so we call this whole process oxidative phosphorylation. But that's all I want you to know, despite the fact that, like we'll see in here in a minute, the book has a lot more detail on some of these pictures than, this isn't a chemistry class, is sort of my attitude about it. Okay, so that was, why is this not? Okay, so that was just a reminder of this piece. All right, so citric acid cycle and electron transport system. Okay. So we talked last time about anabolism and catabolism. And the cells of the body can always go in both directions. They can build things up, they can make larger molecules, and they can break those larger molecules down into smaller molecules, depending on what the cell needs. So when we look at this sort of box diagram, we can see, here's the nutrient pool. So fatty acids, glucose, and amino acids. Those are the basics, right? Those are the building blocks. And the cells can anabolize. They can make bigger things like triglycerides, glycogen, or proteins. And they can catabolize. So we can go back and forth between this complex level and this simpler level. And then from the nutrients, we can also make energy out of those. So we can take all of those nutrients, break them into 2- and 3-carbon chains, feed them into the mitochondria like we were just talking about, and get ATP production, right? And as a byproduct, we get carbon dioxide, which we have to get rid of. And the mitochondrial system only works if there's oxygen present. Because if there's no oxygen present, there's no O negative. So there's no attractor to pull on these hydrogen ions and get that machine to make ATP. So, when you're, like if you have a tissue that's starving for oxygen, this piece gets blocked, the hydrogen ions don't fall, these build up, this stops, so it's like the whole thing backs up in the absence of oxygen. So you have to have oxygen to make that system work. All right, so we can go backwards and forwards, so to speak. All right. Okay, so let's do a few questions here right off the bat. Cells synthesize new organic components. Why? So that's synthesis is usually anabolism, so you know. Did you get the question?
Yes.
Yes? Okay. I don't know if you have until somebody answers, so that's why I ask. All right, good. That is everybody's answer. Okay, so correct answer there is D, all of the above. So check to be sure. Secretion, yes. Most of the things that cells secrete, they have to make those things. So, and most secretions are not nutrient molecules. In other words, there's something else has been built. So yes, that takes energy. Structural maintenance, just like our bones and muscles are never as old as we are, inside a cell, the proteins and carbohydrates that make cells function are constantly being replaced as well. So the only thing in that cell that's as old as that cell is the DNA, because the DNA only gets made when a cell divides. And yes, growth and repair. Cells do grow, and cells get damaged. So the repair system requires synthesis to replace broken parts. All right, the carbon dioxide of respiration is formed during which of those things? All right, last person, go ahead and answer. Oh, must have kicked somebody out. Yeah. Because what happens when you log back in, the number of people in the room changes. So, yeah. All right. Correct answer there is B, the citric acid cycle. And this is an important point because, we all learn when we're in grade school that, we need oxygen and we make carbon dioxide. And once you understand this piece, now you know why those things are true. So the CO2 comes from the citric acid cycle as these hydrogen ions are freed up so they can power ATP production. And the oxygen requirement for respiration is right here, right inside the mitochondria at that electron transport chain. So, some critical things that we do as an organism, we can trace all the way down to that organelle inside the mitochondria. All right. We're going to skip that one. Okay. So the first part of the chapter we just finished is what happens inside the cells. How do cells use nutrients? The rest of the chapter is we're going to look at each of the three classes of nutrients. So sugars, glucose, carbohydrates, fatty acids, and proteins. And we're going to sort of follow them from ingestion all the way through to cellular metabolism. The reason we talked about the mitochondria first is because it's the final common endpoint for a lot of these nutrients. Everything that we take in, or at least everything that we can absorb, can be fed to the mitochondria to create ATP. Now, that's not all that happens to them, but that is, where do they go in the end? They're going to get burned up in that oxidative phosphorylation, what happens inside the mitochondria. All right. So if we look at digestion, first we have the steps and processes that happen in the oral cavity. So in the oral cavity, we get mechanical processing. We're gonna mix up and break up the food that we eat. We're gonna mix saliva into there. Saliva contains two important enzymes that help with digestion. So we're going to talk about those when we get there. Eventually, we swallow, and the material enters the stomach. In the stomach, the saliva enzymes continue to work, at least until they're destroyed by the acid in the stomach. The acid denatures proteins, and protein digestion begins in the stomach. So carbohydrate and lipid digestion begin in the mouth. Protein digestion begins in the stomach. Then in the duodenum, we're going to add the secretions from the pancreas and the liver, which play a role in all three types of nutrients, so carbohydrates, proteins, and fats. So we have that. And then we get into the small intestine, where the bulk of digestion and absorption occur. And then we get to the large intestine, and the large intestine's job is to recover what we put in to make digestion and absorption happen. So as material travels through the large intestine, water and salt are removed, and then eventually what comes out the other end is the indigestible materials. So, and that becomes feces at the end. So the nutrients that come in from the small intestine, they all go to the liver before they go anywhere else. And that's because of the hepatic portal system. The veins that drain the blood from the GI tract are a little different than the rest of the body, in that instead of them just draining back to the heart, they all go through the liver first. So the liver gets sort of first crack at all the nutrients that are coming in. So it can manage the nutrient flow to the rest of the body because of that. And then eventually, those nutrients are going to be put into the bloodstream, and then all the cells of the body are going to take those nutrients out to replenish their own nutrient pool inside. All right? So we're going to talk about carbohydrates first, and then lipids, and then proteins. And we're not going to get to all of this today. But all right, so carbohydrate digestion. So it really is true that of the food that we eat, almost all of it you can divide into carbohydrate, protein, fat. Now, of course, they're all mixed up. In an apple, for example, there's some protein, there's some carbohydrate, and there's very little fat. But in, a hamburger, you've got all three, right? You've got carbohydrate, protein, and fat. It's a rare food that we consume on a regular basis that is only one of those three things. Usually it's their mixtures of things. All right. So carbohydrates. Most of the carbohydrate in our diet are what are called starches. And a starch is a long branching, usually branching anyway, chain of carbohydrates. So in my picture here, each of the circles is a simple sugar, right? So take glucose, for example. Glucose is a 6-carbon ring, and we can chain glucose molecules together. This is relatively small by biologic standards. So we can take this and we can add another sugar to it, right? So we can get a disaccharide, or we can get these long branching chains. So this is what is in the food that we eat. Now, we can also take in simple sugars, but in the common foods that we eat, it's usually starches. So this molecule, way too big to absorb, right? So in order to absorb it, we're going to need to break it into its component pieces, right? We're going to need to take this long branching chain of sugars and turn it first into a double sugar called a disaccharide, but then eventually we want a simple sugar. In other words, just one sugar, not connected to anything else. All right, so that process of carbohydrate digestion starts in the mouth. So in the mouth, there is an enzyme called salivary amylase, and an amylase breaks down starch. So it turns starches into sugars. And usually it's not so good as this. In other words, it doesn't break down the carbohydrate into simple sugars. It usually is more like this, where we have 3 sugars and 2 sugars and 3 sugars and 2 sugars, a lot of di and trisaccharides. That's what an amylase is going to do. So for the so salivary amylase enters with the saliva. So carbohydrate digestion begins in the mouth. And you can sort of experience this. If you take like a cracker, which is practically all carbohydrate starch, and you just put it on the tongue and you let it sit there, eventually the cracker falls apart, right? And that's because of the salivary amylase. Not only is it getting mushy, because of the moisture, but the molecules that make up that cracker are actually being broken down into 2 and 3 carbon chains by salivary amylase. All right, so that process begins there. Then, of course, we swallow, and the stomach doesn't add anything to carbohydrate digestion. But carbohydrate digestion does continue in the stomach because salivary amylase gets swallowed with the food, right? So that process that starts in the mouth continues in the stomach, at least until the stomach pH gets very low. Eventually, salivary amylase gets deactivated by the low pH of the stomach. But in the intervening time, it's working to break down that starch into smaller 2 and 3 sugar pieces. Okay. So then from the stomach, the chyme enters the duodenum, and then in the duodenum, we get another amylase that gets added, and that one is from the pancreas. So now salivary amylase has been destroyed, so we replace it with pancreatic alpha amylase. So one of the enzymes that the pancreas makes digests starch, and it enters the digestive tract at the duodenum. So we get pancreatic alpha amylase added. It's going to do its job as the material passes through the small intestine, and that job is turn starches into 2 and 3 carbon sugars, right? Okay. Or 2 and di and trisaccharides. So 2 sugars or 3 sugars. All right. So then we get to the last step. So the body wants to absorb simple sugars, not disaccharides and trisaccharides. We want single sugars, simple sugars, those are called. So we see what we'll find is a common pattern among carbohydrates, proteins, and fats, and that is this. The last step of digestion happens at the brush border. So you remember that the intestinal epithelial cells have running out of room. Can I erase this? Does everybody have this? Yeah, okay. All right. Okay, so at the intestinal epithelium, so here's a villus, right, a little hill, and then lining that villus were epithelial cells, right, like that, lining all up and down. If we zoom into one of those, it looks like that, right, like Bart Simpson. So this is the brush border. And what we're going to see for proteins, carbohydrates, and lipids is that the last step of digestion takes place in that zone right there. And the reason that happens is attached to this border are enzymes. So there are like these little things sticking out here. I mean, I'm exaggerating the size of it, but so enzymes that are attached to the brush border do the last step. So we're in box 5 here, and so the alpha amylase and salivary amylase has made disaccharides and trisaccharides. Well, at the brush border, attached to that brush border, are enzymes which do the last step, and that is break those bonds between the 2 and 3 carbon sugars to now form simple sugars. All right, so some examples, you do not need to know these, but just to give you an example. Maltose is a sugar, lactose is a sugar, and sucrose is a sugar, right? Sucrose is table sugar. That's the processed sugar that we put in our coffee. So sucrose is actually a disaccharide. It's a glucose and a fructose stuck together. So on the brush border, there's an enzyme that breaks that bond called sucrase. So sucrase breaks sucrose into glucose and fructose. All right, so what do I want you to know? I want you to know that at the enzyme border, or at the brush border, disaccharides and trisaccharides are broken into simple sugars. Okay, so the simple sugars are glucose, fructose, and galactose. So that's what comes in. Now we have finally gotten to the point where the molecule is small enough that it can be absorbed. All right, so the simple sugars are absorbed into the epithelial cells, and then they simply diffuse out into the bloodstream. So what comes from the GI tract, the blood that comes from the GI tract, is chock full of simple sugars, at least when we've had carbohydrates in our diet. And the favorite of the body is glucose, which we're going to talk more about here in a minute. All right, so now we have gone from ingestion all the way to absorption. Okay. Well, what about not all carbohydrates can we digest. For example, cellulose, which is what paper is made of. Trees make cellulose, and we make paper out of it. we don't have the enzymes to digest cellulose. So any cellulose that we eat, it can't get digested, it can't get absorbed because it can't get broken down, so it ends up passing out the other end. So like carbohydrates, indigestible carbohydrates, we call that fiber. We talk about fiber in your diet. Those are carbohydrates that we don't have the enzymes to digest. So things like grass, leaves, wood, we get no nutrition from those things, even though some animals do. The indigestible carbohydrate passes into the large intestine. Now, some of the bacteria in our large intestine can actually utilize some of those indigestible carbohydrates, and they do. And in exchange, they make us those vitamins that we talked about in the last chapter. All right. Okay, so then what happens? So now we've gotten the simple sugars in and into the bloodstream, and they're going to flow to the liver through the hepatic portal system. So that's the big blue arrow here. So this is all those simple sugars coming in. Well, the liver, what we don't want to see, we'll back up, what we don't want to see is we don't want our nutrient levels going up and down all the time, right? Because what the cells of the body need is consistency. But because we don't eat a little all the time, our blood sugar, or the sugars coming into the body, sometimes there's a lot coming in, sometimes there's none coming in. So what the liver does is when there's sugars coming in, it stores those sugars up. When there's no sugar coming in, it lets those sugars out, okay? Because the goal is to keep a glucose level of about 90 milligrams per deciliter, right? So we don't want to see a 300 and a 20. We want a more level curve than that. So the way the liver can store glucose is by making a starch. So what did we take in? Starch. We broke it into simple sugars, and now we're going to reassemble the starch, but we're going to make our own kind of starch, which is called glycogen. So if you take glucose molecules and you string them together in this sort of branching pattern, you get glycogen. So glucose is stored up in long chains as glycogen. And the two principal sites for glycogen storage are the liver and skeletal muscle. And they are stored there for two different reasons. The liver stores glucose as glycogen to keep that glucose level constant. Because the brain, for example, it's the only fuel the brain can use, that neurons can use, is glucose. So we have to have a glucose level. So the liver is doing it for that reason. When there's excess glucose, the liver stores it up, and then when there isn't any glucose coming in, it releases it so that we have a constant level. The skeletal muscles, they store glycogen for a far more selfish reason, and that is for anaerobic contraction. We've all learned about aerobic and anaerobic exercise, right? Well, in anaerobic exercise, when there's when there is more activity than the oxygen can keep up with, skeletal muscle can use glucose to make ATP through the process called glycolysis. So in order to be able to do that, the muscle has to have lots of glucose around. So skeletal muscle stores glucose as glycogen for its own use, for ATP production in anaerobic metabolism. All right, so storage and release. Okay, so glucose level is maintained all the time. Well, the cells of the body can use multiple fuels, except for the neurons, but the rest of the cells can use multiple fuels. So there has to be a way to signal the body with what fuel it should use right now, okay? And one of those signaling molecules you have all heard of, and that is insulin. Insulin opens or closes the glucose door, so to speak, on the cell membranes of cells. So when there's plenty of glucose available, the pancreas secretes insulin. Insulin allows glucose to enter into cells. And once it enters into cells, it can be used either to be stored as glycogen, or it can be metabolized to make ATP using oxygen and creating CO2. All right, so so glucose enters the cell. Glucose is too big to feed to the mitochondria, right? So the first thing that we have to do is we have to break it. So we take our 6-carbon chain here, and we break it here, and we break it here. So we make 2 3-carbon chains out of it. And we're going to talk more about that process here in a minute. All right, so insulin. So here's that insulin receptor, okay, and so on. Let's see. All right. So here we've zoomed in. So here's glucose in the extracellular fluid, right in the blood, and it's going to come into the cell. This is insulin right here, makes this happen, opens this door. Now, once the sugar comes in, it has to get broken up. So we have 6 carbons here, right? And you're looking at this and you're saying, oh my gosh, look at all that chemistry. I don't care for you to know the chemistry here. Okay, I'm not interested in you knowing the chemistry. All I want you to know is glucose comes in. Here, let's use the pointer. Glucose comes in and it gets broken into 2 3-carbon fragments, right? So we're going to skip all this stuff in the middle. In this process, which is called glycolysis, of going from 6-carbon sugar to 2 3-carbon chains, we get some ATP that gets made, but not very much. So if we were to go through all these steps, you'd see that we have to spend a little ATP, but then we make a little ATP along the way. So for each glucose molecule, we get a whopping 2 ATP molecules in this breakdown process. Okay, so that I want you to know. 6 becomes 2 3s. We make 2 ATPs. All right. The other thing I want you to know is this takes place in the cytoplasm. So outside of the mitochondria. This takes place because the 6-carbon sugar is too big to fit into the mitochondria. So this process takes place in the cytoplasm, and it does not require oxygen to work. So in you're lifting weights, right? Real heavy weights, and you're going to hold that weight for a long time. Well, your muscles are going to run out of available oxygen, okay? Which means that they're going to run out of ATP if there isn't some other method. So the other method is this one. Because this process can make ATP in the absence of oxygen, the body can use this in a pinch, so to speak, when there isn't enough oxygen available. Now, it has a couple of downsides, one of which is it causes lactic acid to build up. But what I want you to know, 6 carbons, 2 3-carbons, we make 2 ATP, we don't need oxygen, and this happens in the cytoplasm. Okay? So lots of skipped chemistry. Everybody likes that, right? Okay. Then we take those 2 3-carbon fragments and we feed them into the mitochondria. So even though this looks very messy and nasty, we have already talked about all this, right? I gave you the simple version earlier, yes?
I'm sorry if I'm off topic. Is this also called the Krebs cycle?
Yes.
Okay.
The TCA cycle is called the Krebs cycle.
Okay.
Yeah. Good. Where'd you hear that at?
EMP.
Yeah, okay, yes. That's its older name.
Okay.
We have tried to stop calling things by people's names. So it's like the sphincter of Odie. We don't want to call things after famous physicians anymore, so we change the names. All right. But yes, this is also this part.
It's looking really familiar.
Yes, this part is the Krebs cycle. All right, so ignore all this stuff because I've already given you the simple version that I want you to know. But the 2 red boxes are important. Okay, so for each 6-carbon sugar, we get 2 ATPs from glycolysis, right, out here in the cytoplasm without needing oxygen. And then we get 34 more from what happens in the mitochondria. So you can see that glycolysis is not very efficient compared to the aerobic system, what happens in the mitochondria. So, even though, yes, we can make ATP without oxygen, we make a lot more of it when there's oxygen around. So, grand total, this is the number I want you to know. 36 ATPs are made for each molecule of glucose. All right? So, 36. Why is that important? Because that's pretty significant. One nutrient molecule produces 36 ATPs that can be used for lots of different things. So it's just an example of how efficient the body is at making ATP when there's oxygen around. because the system inside the mitochondria is sort of what makes the whole, we would be far less interesting animals if it wasn't for the ATP production in the mitochondria. All right. All right, so in not just this class, but in your other health care classes, you're going to hear about glucose the rest of your life. And it's because it is the body's simplest and most effective fuel, right? Because cells, even when they're sick, can still use glucose very easily, because glucose is very easy to use. So why glucose? Nothing in biology just happens, right? Evolution has an intelligence to it. So there are a couple of, the reason why glucose is used by the body is because of a couple of different features. One is that it's small and soluble. Glucose dissolves very readily in water, and since most of us, we're mostly water, it means that glucose can travel very easily from place to place in the body. So that's one. Another is glucose, because it's such a stable sugar, when you break it, energy is released. So one of the reasons why we use glucose is because you can make ATP with it, even in the absence of oxygen. So that's another. A third reason is glycogen is a great storage system. If you think about osmosis and okay, let me back up. If you have a ton of glucose, if you kept all that in solution, you'd have lots and lots of dissolved molecules. Anytime you have that, you start to get into an osmosis problem, right? If you pour a ton of salt into the bathtub and you get into the bathtub, it pulls all the water out of your skin, right? You get very pruny very fast. Well, because glucose can be made into glycogen very easily, it means instead of storing, 10,000 glucose molecules, we can make just one glycogen molecule out of those. So we don't have all those pieces dissolved. We have one great big thing. And another thing is glycogen, when it forms very compact granules. So you can store a ton of glucose in a tiny little space because of how glucose becomes glycogen. So that's another reason. And then finally, the fourth reason is glycogen can be turned back into glucose, again, very easily. It takes very little energy to add a glucose to a glycogen, and it takes very little energy to break that bond. Okay, so we can go from glucose to glycogen and back again without having to spend a lot of energy or time or effort. All right, so just some reasons why glucose is the primary energy source. All right. So that's the carbohydrate piece. In some ways, the carbohydrate piece is the easiest piece of the three nutrients. Simple sugar, from starch to simple sugar and absorption. All right. So what about lipids? Lipids are the most challenging nutrient for the body to deal with, and the principal reason for that is the age-old saying that, oil and water don't mix, right? And that creates a number of problems for the system because we're a water-based system, but lipids are a great fuel source. So, how do we handle that? And first, let's look at how do lipids occur in the food that we eat. So most of the lipid in our bodies and in the bodies of the animals that we eat are formed in this fashion. So that is called a triglyceride. So a fatty acid is simply a long chain of carbons, they can get very long, and nothing but hydrogens, right? So that and so on. So this is what I represented here by my squiggly line, right? So a fatty acid is a long chain of carbons with very little oxygen. A carbohydrate, there's an oxygen for every carbon. A fatty acid, there's only one oxygen for this entire molecule, as you'll see in a minute. So it's a different character. And maybe you remember from chemistry talking about polar and nonpolar, right? Polar, carbohydrates are usually polar, which means that you can dissolve them in water. Fatty acids and triglycerides are nonpolar, so they tend to not dissolve in water. So we take these fatty acids and we stick them together, and this backbone right here in red, this is glycerol. And all glycerol does is it connects 3 fatty acid chains, and we call this whole thing then a triglyceride. So what does the digestive tract need to do with this? The cells of the brush border, they can't absorb this triglyceride, it's too big. So what it needs to do is it needs to break 2 of those fatty acids off. So the whole process of digestion here is going to turn that triglyceride into 1 fatty acid, 2 fatty acids, and then what about this thing? We've got a fatty acid with a glycerol attached. This is what we call a monoglyceride. So in the end, the body is going to absorb 1 monoglyceride and 2 fatty acids for each triglyceride that was ingested. Okay? So where and how does that happen? Well, fat digestion begins in the mouth. It's actually the tongue. The tongue creates an enzyme called lingual lipase. And lingual lipase starts the process of turning those triglycerides into fatty acids. So, that occurs, that enters the stomach. And in the stomach, the lingual lipase still is doing its digestion, but the stomach doesn't participate in lipid digestion at all. The stomach mixes everything up, but because oil and water don't mix, what we end up with in the stomach are relatively large droplets of lipid, which creates a problem for fat digestion because of a surface area issue. Okay. All right. So oil and water don't mix. So what we end up with are droplets, right? The enzymes that are going to break up that triglyceride, they're out here, right? They're in solution. They're in water. So this is oil. So the only way that these enzymes can get at this triglyceride is only at the surface, right? So if we have a few large droplets, the only activity is going to be at the surface here. What we're going to see is that what the body does is turn that big droplet into smaller ones, and now instead of having to work your way in, this droplet has been broken up and it can be worked on in more places, right? So it can be worked on faster. So that piece occurs in the duodenum. So in the stomach, we get no contribution. Lingual lipase continues to work until the acid destroys it. In the duodenum, we get this emulsification. So what does this? Large droplets become smaller ones. Those are bile salts. So in the bile that's secreted by the liver are molecules that look very, very much like soap molecules do. Soap molecules are special because they have a polar end and a non-polar end. So they essentially can take a large droplet of oil and break it up into many smaller droplets. I'm not going to go into how that happens. Just know that that's what happens. We call that process, this is emulsification, where we take a fat and we break it up into many smaller droplets so that it can mix more easily with water. All right? So, and in our systems, the emulsifier are bile salts found in the bile. Okay, so we do that bit. These smaller droplets get special names. We call those micelles. That word is right here. Micelles are very tiny droplets that result from emulsification. All right. It's the same thing that happens in your dish, when you're doing dishes. When you add a little dish soap, that film of oil on the top disappears. Well, it didn't disappear. It got broken up into many smaller droplets so that it can get washed away with the water more easily. All right. The other important piece that happens in the duodenum is the pancreas adds its lipase, pancreatic lipase. A lipase breaks down fats, lipids. ASE means to break. So like an amylase breaks down amylose, which is a starch. A lipase breaks down lipids. So the pancreas releases its lipase into the duodenum, and it is the most important lipase in the whole system. So the pancreas's contribution to fat digestion is critical. If your pancreas is sick, you won't digest fats very well, or at all, because there aren't very many places in the body where lipases are secreted. All right, so pancreatic lipase. And then when you think lipid, think cholecystokinin, because that hormone, CCK, is released in response to lipids being in the digestive tract. And CCK causes the gallbladder to contract, which makes sense because we need bile to make emulsification happen, right? And then CCK also triggers the pancreas to release its pancreatic lipase. All right. So that's box 3. Okay, then, so eventually we're going to get that digestion that's going to take place. So now we're going to zoom in and we're going to look at the brush border again. So what happens there? Well, eventually those triglycerides become monoglycerides and fatty acids. And so here's a micelle, a tiny little bubble, right? It comes up to the membrane, and then what's inside it, monoglycerides and fatty acids, are absorbed into this intestinal epithelial cell, and then the bile salts are released. Instead of taking the bile salts in, the bile salts are let go so that they can go and do more work, can go and emulsify some more. So the monoglycerides and fatty acids come in, and then they're remade into a triglyceride. So before they even leave this cell where they entered the body, they're going to get repackaged as what they started as. So you might say, well, why do we break it down then? Why don't we just absorb triglycerides? Nobody knows. Too big maybe? But the first thing that happens right here is the monoglycerides and fatty acids are reassembled into a triglyceride. And now we have a transport problem. Triglycerides don't mix with water, right? We were just talking about that. So how are we going to move them in the blood? And the body has a system for that, and it's called the lipoprotein. So if the triglyceride won't dissolve in water, one thing you can do is you can package it. So if you take a bunch of triglyceride here, if you tried to put that in the bloodstream, it wouldn't move because it's not soluble. But if you wrap it in a protein that is water soluble, now you can move this thing around. So this protein package with triglyceride inside, we call this a chylomicron. And it's the largest and first of the lipoproteins, right? So it's chylomicrons with their triglycerides inside. They don't go into the blood. Instead, they go into the lymph, right? In that central lymphatic of the villi called a lacteal. So they're absorbed, emulsified, absorbed, repackaged as triglycerides, wrapped in protein, released from the cell. They enter the lymphatic system, and then they're going to go and up the thoracic duct in the center of the chest, get dumped into the bloodstream, and then they make their way back to the liver. And that's where we're going to stop for today. So we'll pick up the story of the lipoprotein on Monday.